Welcome to this explainer. Okay, we are jumping right in today because we're looking at one of the absolute busiest, most vital organelles in all of human biology. Inside every single eukaryotic cell in your body, there's this logistical masterpiece that runs 24/7. No sleep, no breaks. Without it, your cells would descend into total chaos in a matter of minutes. So today, we're unpacking exactly how this microscopic marvel works and how scientists are actually learning to hack its machinery to cure disease. If you take away just one single concept from our entire explainer today, let it be this. The Golgi apparatus is basically the cell's molecular post office. Think about how a real post office works, right? You drop off a generic cardboard box. The postal workers tape it up, slap a specific barcode on it, sort it by zip code, and throw it on the exact right truck so it gets where it needs to go. Well, the Golgi does the exact same thing, just with proteins and lipids. It takes raw cellular materials, chemically tags them, and routes them to their final destination. So formally speaking, the Golgi is a stack of flattened, membrane-bound sacs. In biology, we call these sacs cisternae, and honestly, they look a lot like a stack of deflated pancakes. But don't let the look fool you. This is a high-efficiency processing plant. Since its entire job is packaging and shipping, you're going to find really well-developed Golgi apparatuses in cells that do a lot of secreting. We're talking pancreatic cells pumping out digestive enzymes, plasma cells firing off antibodies, or goblet cells releasing mucus. To really get the Golgi, it helps to look at its neighbor, the rough endoplasmic reticulum, or rough ER. Think of the rough ER as the manufacturing plant. It's covered in ribosomes, which are those tiny machines that actually synthesize the raw proteins. But here's the major difference. The Golgi doesn't have any ribosomes at all. It doesn't make the proteins. Instead, it takes the raw, initially folded proteins from the ER and does some heavy-duty modifications. The ER sends the cargo, and the Golgi sorts it and ships it out. It's manufacturing versus distribution. And this dynamic really highlights another crucial relationship, the Golgi and the lysosome. Now, the lysosome is basically the cell's recycling center and garbage disposal, right? Its whole job is to digest and degrade waste. But how does it get those highly destructive digestive enzymes in the first place? Well, the Golgi actually creates the specific molecular signals that route those dangerous enzymes safely to the lysosome. Without the Golgi perfectly packaging everything, the lysosome just couldn't do its job, and those enzymes could actually end up accidentally destroying the cell itself. So how is this microscopic post office actually laid out? Let's take a quick structural tour. The Golgi is highly polarized, which just means it has a very distinct front, middle, and back. These are divided into three functional zones. First up, we've got the cis-Golgi network, which acts as the receiving dock. Then we move into the middle Golgi, the busy processing floor. And finally, we end up at the trans-Golgi network, which is the shipping department. Let's start with Section 1, the receiving dock, also known as the cis-Golgi network. Proteins don't just randomly float over from the ER to the Golgi. They arrive safely packaged inside specialized delivery trucks called transport vesicles. Specifically, these inbound vesicles are coated in a protein called COPI-2. You can picture COPI-2 as an armor-plated delivery truck pulling right up to the cis-Golgi receiving dock. The vesicle fuses with the Golgi membrane, drops off its cargo of fresh proteins and lipids, and boom, the biological sorting process kicks off. Moving right along to Section 2, the processing floor, or the medial Golgi. Now, there is a massive variety of modifications happening right here on the processing floor. The most prominent one is called glycosylation. This is where carbohydrate groups, basically complex sugars, are added to or modified on the proteins. And this is a total game changer, because it alters how the protein folds, how stable it is, and even how it interacts with other cells. We also have phosphorylation, which adds phosphate groups, sulfation for sulfate groups, and proteolytic processing, where inactive proteins are literally snipped and cleaved into active hormones. It is customizing raw proteins at unbelievable speeds. An absolutely crucial point here is how the Golgi uses these chemical modifications to direct traffic. When the Golgi gets a raw enzyme that's meant for the lysosome, it does a very specific phosphorylation. It tags it with something called mannose 6-phosphate, or M6P for short. Think of this M6P tag as a literal molecular barcode. It ensures this dangerous digestive enzyme gets sorted perfectly and never gets lost in the mail. Because if it didn't get this tag, that enzyme would be mistakenly secreted right outside the cell. Next up is Section 3, the shipping department, the trans-Golgi network. Once we're at the transphase, the finished proteins are sorted into outbound transport vesicles based on those exact chemical tags we just talked about. and they've got a few main delivery routes to take. Some head straight to the plasma membrane to help the cell grow or remodel its surface. Others, carrying that M6P tag, are dispatched directly to the lysosomes. And a lot of them are packed into secretory vesicles to be dumped outside the cell entirely. Oh, and just like copy I brought things in, a different vesicle-coating protein called clathrin helps package and guide these outgoing shipments from the trans-Golgi to their final stops. Okay, let's put this all together into one epic journey. We call this the secretory pathway. Imagine a single molecule of insulin. It starts as a blueprint in your DNA, gets transcribed to mRNA, and then a ribosome reads it and synthesizes the raw protein right into the rough ER. From there, a transport vesicle carries it to our molecular post office, the Golgi. The Golgi processes it, perfects it, and loads it into a secretory vesicle that merges with the plasma membrane, releasing that insulin into your bloodstream to regulate your blood sugar. It's a flawless, multi-organelle relay race. But as we know, biology is fragile. What happens when this exquisite machinery breaks down? What happens if the cell's molecular post office starts slapping the wrong tags on the wrong packages, or its shipments just stop moving entirely? The stakes here aren't just cellular, they're systemic, and it can quite literally be a matter of life and death. In fact, one of the most intense areas of current research is the Golgi's role in cancer. Cancer cells actually hijacked the Golgi apparatus. By altering those complex glycosylation pathways on the processing floor, a tumor cell can completely change its outer carbohydrate shell. This abnormal glycosylation lets the cancer cell evade our immune system's radar. It even alters cell adhesion, which makes it easier for the tumor to break apart, invade other tissues, and metastasize. Plus, all that disrupted vesicular trafficking just fuels the tumor's uncontrolled growth. And beyond cancer, there are inherited genetic mutations that directly impact the Golgi's ability to glycosylate proteins. We call these congenital disorders of glycosylation. Because this protein processing is so incredibly fundamental to every single cell type in your body, even a tiny mutation in the Golgi's machinery results in devastating systemic disorders right from birth, affecting the brain, muscles, and multiple organ systems. It really highlights just how much we rely on this microscopic sorting center. But it's not all bad news. Let's look at Section 4, Hacking the Post Office, the Golgi in Biotechnology. You see, scientists aren't just watching these failures happen, they're learning to take control. Modern biopharmaceutical manufacturing actually relies entirely on hacking the Golgi. When we want to mass-produce therapeutic hormones, vaccines, or highly targeted monoclonal antibodies, we have to use eukaryotic cells. Why? Because we need their Golgi apparatuses. We need the Golgi to correctly glycosylate these therapeutics. Because if those carbohydrate structures aren't perfect, the drug might be completely unstable or even cause a severe immune reaction. Scientists are even practicing glycoengineering right now, literally customizing the Golgi's pathways to build entirely new, improved therapeutic proteins from scratch. So, as we wrap up our tour today, I want you to remember this simple flow. The endoplasmic reticulum builds it. The Golgi modifies it, sorts it, and packages it, and then it's shipped to its final destination. It's an elegant, continuous pipeline keeping every complex organism alive and functioning. ER, Golgi, modification, sorting, packaging, destination. Keep that in your head. And that leads us to a really thrilling frontier. Right now, modern research is actually combining Golgi biology with artificial intelligence to predict exactly how proteins are trafficked and modified. It begs a massive question. With AI accelerating our understanding like never before, what currently incurable diseases will we conquer once we finally master the cell's molecular post office? We're just scratching the surface of what's possible in organelle dynamics here. Thank you so much for joining me on this explainer. Keep asking questions, and I'll catch you on the next one.